Imagine if we could fix broken genes the same way we edit a typo in a document. But here's the shocking reality. Until now, getting those genetic editing tools to where they need to go has been medicine's most frustrating roadblock. Breaking news from the scientific world just changed everything. Researchers have cracked the code on making CRISPR gene editing 3 times more powerful than ever before. But here's where it gets controversial. This breakthrough might completely reshape how we think about treating genetic diseases, and not everyone in the medical community is ready for what comes next. The game-changing numbers that have scientists buzzing. Let's talk about what 3 times more effective actually means in real terms. The new delivery method doesn't just improve CRISPR slightly. It creates a dramatic leap forward that could transform genetic medicine as we know it. Gene editing machinery now enters cells with triple the success rate compared to current methods. The system cuts toxicity dramatically while boosting editing efficiency by 300%. Precise DNA repairs became more than 60% more successful. The technology works across multiple human and animal cell types. But here's the part most people miss. This isn't just about making CRISPR work better. It's about making it work at all in situations where it previously failed. Why CRISPR has been medicine's most promising yet frustrating tool. Think of CRISPR as the world's most precise molecular scissors. It can cut out disease-causing genetic defects, insert healthy DNA sequences, and even add entirely new cellular functions. The potential is mind-blowing. We're talking about possibly curing genetic diseases that have plagued humanity for millennia. Here's the catch that's been driving scientists crazy. CRISPR is like having the perfect key to fix genetic locks, but no reliable way to get that key to the right door. The gene editing machinery simply cannot enter cells on its own. It always needs a delivery vehicle. Much like how a surgeon needs to get into an operating room before performing surgery. Current delivery methods have been frustratingly inadequate. Scientists typically rely on 2 main approaches, both with serious drawbacks. Viral vectors work efficiently because viruses naturally excel at sneaking into cells. That's literally what they evolved to do. However, our immune systems recognize these viral invaders and often launch aggressive responses that can cause painful, sometimes dangerous side effects. It's like using a burglar to deliver your mail. Technically effective, but you might not like the consequences. Lipid nanoparticles represent the safer alternative. Think of the same technology used in COVID-19 vaccines. But they're disappointingly inefficient. These particles frequently get trapped in cellular compartments called endosomes, where they sit uselessly, unable to release their precious CRISPR cargo. Imagine having a locked briefcase full of life-saving medicine, but losing the key. That's essentially what happens. Enter the revolutionary DNA-wrapped taxi solution. Scientists at Northwestern University just unveiled something that sounds like science fiction, but works like precision engineering. They've created what could be described as molecular DNA-wrapped taxis called lipid nanoparticle spherical nucleic acids, or LNPSNAs, for those who prefer shorter names. Here's how these tiny marvels work, explained in terms anyone can understand. Picture a protective sphere roughly 50 nanometers in diameter. That's about 1,000 times smaller than the width of a human hair. Inside this sphere sits the complete CRISPR toolkit, Cas9 enzymes, the molecular scissors, guide RNA, the GPS system that tells CRISPR where to cut, and DNA repair templates, the replacement parts. But here's the ingenious part. The entire package gets wrapped in a dense, protective shell made of DNA strands. This isn't just protective packaging. It's a sophisticated delivery system. The DNA coating serves multiple crucial functions. It shields the delicate CRISPR machinery from being destroyed during transport. It acts like a molecular postal code, determining which organs and tissues the particles travel to. It makes cell entry dramatically easier by interacting with surface receptors that cells naturally recognize and welcome. Chad A. Merkin, the Northwestern researcher leading this breakthrough, explains it perfectly. Simple changes to the particle structure can dramatically change how well a cell takes it up. The SNA architecture is recognized by almost all cell types, so cells actively take up the SNAs and rapidly internalize them. The controversial question everyone's asking. Now here's where things get interesting and potentially controversial. If this technology is as revolutionary as the data suggests, we might be looking at a future where genetic diseases become as treatable as bacterial infections. But that raises some profound questions that society isn't necessarily prepared to answer. Should we be editing human genes on this scale? Who gets access to these treatments? And perhaps most controversially, if we can fix genetic defects, who decides what constitutes a defect versus natural human variation? Merkin doesn't shy away from the implications. CRISPR is an incredibly powerful tool that could correct defects in genes, to decrease susceptibility to disease, and even eliminate disease itself. But reaching and entering the right cells, and the right places within those cells, requires a minor miracle. And this is the part most people miss. We might have just witnessed that minor miracle become reality. Real-world testing results that speak volumes. The Northwestern team didn't just create these LNP SNAs and call it a day. They put them through rigorous testing across various human and animal cell types, including skin cells, white blood cells, human bone marrow stem cells, and kidney cells. The results were consistently impressive across every metric that matters. Cellular uptake increased up to 3 times compared to standard lipid particle delivery systems. Toxicity dropped significantly, addressing one of the biggest concerns with current methods. Gene editing efficiency tripled across the board. Precise DNA repair success rates jumped by more than 60%. What makes these results particularly exciting is their consistency. This wasn't a case of the technology working well in one specific cell type but failing in others. The improvement was universal, suggesting we're looking at a fundamental breakthrough rather than a narrow technical advancement. The bigger picture, structural nanomedicine, revolution. This breakthrough represents more than just an improved CRISPR delivery system. It validates an entirely new approach to medicine called structural nanomedicine. The idea that how we structure our therapeutic delivery systems is just as important as the medicine itself. Think about it this way. The same medication can have completely different effects depending on how it's packaged and delivered. A pill works differently than an injection, which works differently than a patch. The LNP SNA system takes this principle to the molecular level, demonstrating that the architecture of nanoparticles can dramatically influence their therapeutic effectiveness. Merkin, who pioneered this field, puts it in perspective. By marrying 2 powerful biotechnologies, CRISPR and SNAs, we have created a strategy that could unlock CRISPR's full therapeutic potential. What happens next? The clinical reality check. Before anyone gets too excited about immediately available genetic cures, let's address the elephant in the room. We're still in the early stages of development. The current results come from laboratory cell cultures, which, while promising, represent just the first step in a long journey toward human treatments. Merkin's team is already planning the next phase, validation in living disease models. This will provide crucial data about how the system performs in complex biological environments, not just isolated cell cultures. Interestingly, Northwestern has already spun out a biotechnology company called Flashpoint Therapeutics, which is working to commercialize this technology and move it toward clinical trials. 7 SNA-based therapies are already in human clinical trials for various conditions, including a Phase 2 trial for Merkel cell carcinoma. The platform advantage. One solution, many applications. One of the most exciting aspects of this breakthrough is its modular nature. The LNP SNA platform can potentially be adapted for a wide range of genetic conditions and therapeutic applications. This isn't a one-trick pony. It's more like a Swiss Army knife for genetic medicine. The implications stretch far beyond single gene disorders. We could be looking at new treatments for complex diseases like cancer, neurological conditions, and even aging-related genetic damage. The versatility of the platform means that once researchers perfect the delivery system, they can potentially adapt it for numerous therapeutic targets. The uncomfortable truth about medical innovation. Here's something that might make you uncomfortable, and it should. Every major medical breakthrough forces us to confront difficult questions about access, ethics, and the future of human health. This CRISPR advancement is no exception. If genetic editing becomes as routine and effective as the data suggests, we'll face unprecedented decisions about human enhancement, genetic equality, and the definition of normal. Human variation. These aren't just scientific questions, they're societal ones that will require input from ethicists, policymakers, patients, and communities worldwide. Some might argue that improving genetic medicine is an unqualified good. After all, who wouldn't want to eliminate genetic diseases? Others worry about creating a world where genetic perfection becomes expected or where access to genetic improvements creates new forms of inequality. What this means for you and your family. While widespread clinical applications are still years away, this breakthrough represents a fundamental shift in what's possible with genetic medicine. For families affected by genetic disorders, it offers genuine hope for treatments that were previously impossible to deliver effectively. For the broader medical community, it demonstrates that sometimes the biggest advances come not from discovering new drugs, but from finding better ways to deliver existing ones. The principles behind this research could revolutionize how we approach not just genetic diseases, but potentially many other medical conditions. The bottom line, a new chapter in medical history. We may be witnessing the beginning of a new era in genetic medicine. One where the promise of CRISPR finally matches its practical capabilities. The threefold improvement in effectiveness isn't just a statistic. It represents the difference between experimental hope and clinical reality for millions of people worldwide. Merkin's final observation captures the significance perfectly. CRISPR could change the whole field of medicine. But how we design the delivery vehicle is just as important as the genetic tools themselves. Now I want to hear from you. Do you think we're moving too fast with genetic editing technology, or not fast enough? Should society be more concerned about the ethical implications or more excited about the therapeutic potential? And perhaps most importantly, if this technology could prevent genetic diseases in your family, would you use it? Share your thoughts in the comments below. This is a conversation that affects all of us, and your perspective matters in shaping how we navigate this new frontier of medicine.